Now we begin our study of the muscular system. On the most fundamental level, muscles contract. Let's go over a few other functions. You all know that muscle contraction produces motion, enables us to walk, but there's also lesser known functions as well, like muscles enable us to maintain body posture. For example, even when a muscle appears to be at rest, some muscle fibers are actually experiencing sustained contraction. It's what we call muscle tone. And muscle tone originates from signals in the spinal cord that are sent to muscle fibers so they can contract. But if this tone is lost, then what happens is someone, if the tone is lost because if someone loses consciousness, they don't have that sustained contraction anymore, and they'll pass out or fall over. Muscles also support soft tissue. What I mean by that, like your abdominal wall keeps your viscera or your visceral organs from falling out anteriorly. We also have this group of muscles together makes up what's called the pelvic floor, and that keeps your visceral organs from sort of falling down, if you will. Muscles also regulate the entering and leaving of material. I guess this is best illustrated talking about the circular muscles that like the sphincters that operate around the orifices of your urinary tract and your digestive tract. So it regulates the leaving of materials like solid waste and also urine, and also what comes into your body, your mouth. Maintain body temperature. Just like when you put gasoline in your engine, that gasoline is used to make your car go forwards, but your engine also gets hot because some of that gasoline is converted to heat. Same thing is true in your body. Your muscles aren't 100% efficient either. And what is the energy that's not used for contraction is then dissipated as heat, and that heat is used to keep your body warm. In fact, that's the primary mechanism through which your body maintains thermal homeostasis. Let's look at the structure and organization of a muscle. We're going to go through this fairly quick. You don't need to know all of these. I'm just going to highlight it, and I'll tell you what you need to know in a moment. So this is your femur, and attached to the greater trochanter, we have this tendon, and this tendon is covered with fascia. And then if we look in closer, we have an epimysium, a perimysium, and even an endomysium. A lot of this is dense connective tissue, and this dense connective tissue surrounds not only these big fascicles of muscle, but also the individual cells, and then those eventually come together and they form the tendon. And then the tendon connects to the bone. These fibers are interwoven with the periosteum of the bone. So that's how muscle physically connects to the bone. If you could take a cross-section of a muscle, we see all these little things called fascicles. And if you look at one of these fascicles are composed of fibers. A fiber is another name for a cell. That's something I want you to know. Because when we say muscle fiber, what we're really referring to is a single muscle cell. And some of these muscle cells are very long. They run the entire length of the muscle. And then inside the muscle fiber, we have these things called myofibrils. And these myofibrils are composed of filaments, actin and myosin. And that's what's responsible for muscle contraction. So let's look at that now. We're looking at one muscle fiber, one cell. And here's one of the myofibrils. And notice that the myofibrils are composed of these repeating units called sarcomeres. This is a word I want you to know, so write that down. A sarcomere goes from this, which is called the Z line, to this Z line. Here's another sarcomere from here to here. The sarcomere is the functional unit, like the functional contractile unit of a muscle. And there's a lot going on, but I want you to know these two proteins. There's myosin, which is a thick filament shown in purple. And there's also actin, which combined with a couple other proteins make up what's called the thin filament, which is shown in blue. So let's look at one of these sarcomeres. So here's a sarcomere under a microscope. It's going from this dark line to this dark line. That's Z line to Z line, which is what we see here. So notice where we only have the thin filaments, the actin, the banding appears light. But here where we have actin and myosin together, it appears darker. And here we have only myosin. But I think the takeaway here, what we want to walk away with is this repeating pattern of light and dark, that banding produces the striations that we see in skeletal muscle. And it's these actin and myosin fibers, when they slide past one another, that creates muscle contraction. And we call that the sliding filament theory of muscle contraction.
Sliding filament theory is the mechanism by which muscles are thought to contract at a cellular level. An understanding of the structure of skeletal muscle is useful when learning how sliding filament theory works. Each muscle is made up of a number of bundles of muscle fibers. Each bundle of muscle fibers contains anywhere from 10 and 100 individual fibers. Each muscle fiber itself contains cylindrical organelles known as myofibrils, which themselves are bundles of proteins called actin and myosin. Surrounding the myofibril, there is a network of tubules and channels called the sarcoplasmic reticulum, where calcium is stored. Each myofibril can be broken down into functional repeating segments called sarcomeres. If we look at a two-dimensional model of a sarcomere, it consists of actin and myosin. When a nerve impulse arrives at the muscle, it causes a release of a chemical called acetylcholine. The presence of acetylcholine causes depolarization, enabling calcium to be released from the sarcoplasmic reticulum. The calcium binds to troponin, changing its shape and so moving tropomyosin from the active site of the actin. The myosin filaments can now attach to the actin, forming a cross bridge. The breakdown of ATP releases energy, which enables the myosin to pull the actin filaments inwards, contracting the muscle. This occurs along the entire length of every myofibril in the muscle cell. When an ATP molecule binds to the myosin head, the myosin detaches from the actin and the cross bridge is broken. When the ATP is then broken down, the myosin head can again attach to an actin binding site further along the actin filament and repeat the process. This repeated pulling of the actin over the myosin is often known as the ratchet mechanism. This process of muscular contraction can last for as long as there are adequate ATP and Ca+ stores. Once the nerve impulse stops, the Ca+ is pumped back to the sarcoplasmic reticulum and the actin returns to its resting position, causing the muscle to lengthen and relax.
We saw what happens inside the cell when the muscle contracts, but what causes the muscle to contract? We need to look at the synapse between a motor neuron and a muscle fiber. That's called the neuromuscular junction. So here we're looking at this motor neuron, and here's an impulse that travels through that motor neuron, and then when this impulse gets to the muscle fiber, it's that impulse that causes or initiates the contraction within the fiber. So the muscle fiber contracts when stimulated by the motor neuron. Let's look more closely at the neuromuscular junction. The motor neuron communicates with the muscle fiber using a neurotransmitter called acetylcholine. So remember that name. We're going to look at that in just a moment. So here's a motor neuron, and this is the cell membrane of the muscle fiber. This is the neuromuscular junction. If we can zoom in on that neuromuscular junction, we see that this is the motor neuron, distal end of it, and this is the cell membrane of the muscle fiber. Notice there's no physical connection between the motor neuron and the muscle fiber. There's only going to be chemical communication. So these vesicles here are filled with acetylcholine, and when the nerve impulse propagates through here at the end, these vesicles then release the acetylcholine into the synaptic cleft, and the acetylcholine binds with the cell membrane, and then the contraction then is initiated from there. So let's look at that. Here's the acetylcholine being released into the synaptic cleft. We see the depolarization of that membrane, and that's going to propagate through. That's the endoplasmic reticulum. And then that releases calcium ions. Those calcium ions expose the binding site on actin, and then these myosin heads are able to bind with the actin, and that's and then it pulls on it, and that's the contraction. That's the sliding filament theory.